real deal with them. So meiosis is a little different because, let's say, here's your maternal, here's your paternal copy. This is doubled. This is doubled once, but then with meiosis, it happens again. So right here you have two copies, and that's what that 2n stands for. But over here, you only have one copy of a chromosome going into 4 cells. And let's say this one is the maternal one, this one's paternal, let's say this one's the paternal from here, and this one's the maternal. So this is the difference with meiosis, and remember that's because the sex cell is going to go combine with the male or female sex cell, depending on whose this is, and that will form a 2n. So a 1n, say from the male, plus female. This is the zygote. This is where you have 23 full pairs of chromosomes. So these don't have 23 pairs, these just have 23 chromosomes. That's it, no pairs. And this is 2n, and this is the zygote. So there's 3 main sources of variation during meiosis, and you can take a closer look like in your textbook for this. So the first one, I think this happened first, is crossing over. So you have your maternal and your paternal. So when these chromatid pairs are ready, so I'll do it like this. And remember, these are the same chromosome, and they're ready to be copied, but it's just maternal copy and a paternal copy. This is where your alleles come from. This is like Mendel and stuff. If you, this might actually be easier if we just do one. There's a process in meiosis where breaks are initiated in the chromosomes, and then they ultimately kind of go together. Here, we'll do this. And so what happens, say, is that there's a break here. Let's see if I can draw this. And it would reattach, it would like, oops, sorry, it would go over here. It would kind of switch. And this recombination can cause like shared genetic information. And this has to always be on the same chromosome, because if you put DNA from another separate chromosome, like say this is chromosome 6, it could never go over to like chromosome 8. That would mess everything up. So this is just between maternal and paternal copies. So say that they switch like this. Have your maternals. But in this case now, the maternal actually has a bit of that paternal chromosome now, and they've kind of switched. They've gained regions from each other. And so now, when this splits into 4, for example, here, let's go. Sorry, this is kind of a lot. So there's going to be 4 chromosomes distributed through meiosis here. So after the crossing over, you've got 1 that ended up with the maternal. You've got 1 that ended up with the maternal paternal side. Yep. So you don't have to put that though. You've got 1 with paternal, and it's just this one, it's just regular paternal. And then the paternal one that has the little variation from the mother's one. So you can see here, these 2 are more varied, for example. So when these go on and fuse with the partner haploid cell, the 1n cell, they can cause that variation. They can have that genetic variation. So the other thing is that when a chromosome or when a cell is dividing and you're choosing which one, let's say during meiosis, to send the paternal or the maternal ones, when they long align the metaphase plate, let's say maternal is in red, paternal is in black, let's say. I think I had that reverse on the other one. say maternal's in black, red is paternal. So you need to align pairs of chromosomes, but you don't necessarily need to make them go on the same side. So when these are split from each other, this cell is going to have these chromosomes, so it would have 1, 2, 3 from the paternal. Let's call this chromosome 1, 2, 3, 4, 5, 6. And these are like the actual chromosome numbers. But then this cell over here is going to have these copies. And you can't control which side it flips on or which side the maternal or the paternal are going to go on in a certain haploid division. And the other source of variation, so that was number 2, was independent assortment. So crossing over. Independent assortment. And the last one is that it's just large number. When there are tons of these haploid cells after, let's say, a female, so these are male, let's say this is female egg has been kind of selected after a large process and it has a lot of variation, but then there are hundreds of thousands of male haploids that contribute to the variability of what the ultimate zygote, the little 2N, like the fertilized egg, is going to actually become. So that's kind of the brief overview. You'll get to kind of visualize some of this in lab. Sorry if this video was a little dense. It's definitely like kind of a lot in this one, but we should be good for lab and should be able to like clarify face-to-face some of this. So let me know if you have any questions, and I'll see you on Friday.